Everyone, welcome back to my educational channel on biology. Teacher Janet here. How are you doing with your revision? On the request of some students who want to know more and understand more about this chromosomal mutation, I've decided to produce this video. All right, and this is from chapter 12 of Form 5 syllabus. All right, and it's quite important for the SPM as the only year that's been asked is in the paper 2 is in 2021, all right, where two diseases were compared. So let's find out more about chromosomal mutation, especially if you need to do some last minute revision. Okay, you can follow along here. So what is mutation? First of all, it is a spontaneous and a random change in the genetic material. Okay, in the genetic material, an abnormal change in the genetic material, such as the DNA or chromosomes in a cell. Okay, and chromosomal mutation involves two types: changes in chromosomal structure or changes in chromosomal number. Okay, these are all abnormal changes. Now, in the past video, we have discussed gene mutation, which is different. Okay, so later we'll do a comparison of gene mutation and chromosomal mutation. So, what is an example of changes in chromosomal structure? Now, for example, maybe the person is exposed to mutagens like ultraviolet rays, excessive ultraviolet rays, or food pesticides, all right, benzene and so forth, and that may trigger the changes to occur, okay, the mutation to occur. So as an example of changes in chromosomal structure, for example, a chromosome here may have the segments P, Q, R, S, T, U, but maybe the segment, the segment called Q it breaks off and is removed forever, okay? Then the two ends join back together. So now the chromosome becomes shorter. So this example of deletion, which is a type of chromosomal mutation here, right? And can give rise to diseases like Cri-du-Chat syndrome, okay? Now, in the second type, changes may occur in the chromosomal number. For example, in Down syndrome child, each diploid cell has 3 chromosomes, number 21, all right, instead of 2. So this will cause the Down syndrome child to have 47 chromosomes in each diploid cell because of the extra chromosome 21. Okay, so let's find out more about the different diseases caused by changes in chromosomal structure and chromosomal number. The learning standards are as follows. Number 1, describe with examples the two types of mutations. Number 1 is gene mutation, which was covered in the previous videos, and then number 2 is chromosomal mutation, which I'm going to discuss in this video. For gene mutation, examples of diseases caused by gene mutation are sickle cell anemia, albinism, thalassemia, cystic fibrosis, and hemophilia. Think of the acronym CHATS. C for cystic fibrosis, H for hemophilia, A for albinism, T for thalassemia, and S for sickle cell anemia, right? Now, let's go on to chromosomal mutation. So in this video, we'll discuss diseases caused by chromosomal mutation, such as Down syndrome, Turner syndrome, KLINEFELTER syndrome, Jacob syndrome, and Cri-du-Chat syndrome. So notice all of these caused by chromosomal mutation. All these diseases have the word syndrome in it. Now, the acronym memory tip, which is very useful, is King, Jacob, Turn, Down, Chris. King for KLINEFELTER syndrome, Jacob for Jacob syndrome, Turn for Turner syndrome, Down for Down syndrome, and Chris for Cri-du-Chat syndrome. All right. So we also discuss the different types of chromosomal mutation. Then number 2, relate mutations of somatic cells and gametes to variation. All right. So this will also be discussed. Now, so here in this picture, you see chromosomal mutation. Example number 1 is duplication. Okay, just now I discussed deletion. And then chromosomal mutation number 2, example 2 is when there's a change in chromosomal number, such as KLINEFELTER syndrome, where the patient has 3 sex chromosomes in each diploid cell, XXY. Here's the concept map on genetic factors that cause variation. So factors that cause variation can be divided into genetic factors and environmental factors. Now, for genetic factors, there are lots of things to talk about. Okay, the content is a lot. So genetic factors that cause variation are A, crossing over and independent assortment of chromosomes. All these occur during meiosis. Okay, and then random fertilization. all right? And then B mutation is the other genetic factor. So there are 4 genetic factors: crossing over, independent assortment of homologous chromosomes, random fertilization, and mutation. All these are genetic factors that cause variation. Now, under mutation, we have discussed what is a mutagen, which is a substance that can cause mutation or spontaneous and random changes in the genetic material of a cell, right? So there are 2 types of mutations: gene mutation and chromosome mutation. And then gene mutation, examples of diseases that are caused by gene mutation: sickle cell anemia and albinism. So here we have discussed in another video about the gene mutation, which are actually changes in nucleotide base sequence of DNA or genes. And there are 3 types of gene mutations that we discussed. Base deletion, that means the nitrogenous base like A, T, C, and G. So base deletion. So thalassemia is an example of a disease caused by base deletion. Base insertion, base substitution. All this has been discussed. Now we are going to talk about chromosomal mutation. All right. So the 5 diseases that will be discussed are Down syndrome, Cri-du-Chat syndrome, Turner syndrome, Klinefelter syndrome, and Jacob syndrome. Now I will give you the acronym for this, and also we will try to remember get some memory tips to remember the facts about these syndromes. Okay, so continue to view, and then you will see the memory tips on all these, which will help you to remember all these syndromes. So as we said, chromosome mutation, there are 2 types. The first type is when there's a change in the number of chromosomes, and the second type when there's change in structure of chromosomes. Okay, so we will discuss Down syndrome, Turner syndrome, Klinefelter syndrome, and Jacob syndrome. And also for change in structure of chromosomes, we will discuss deletion that causes Cri-du-Chat syndrome, inversion, duplication, and translocation. All right. So before we start, let's just go through this question, which can be asked in the SPM. Compare and contrast gene mutation and chromosomal mutation. So gene mutation has been studied earlier, right? Now, this can be 4 marks or 6 or 8 marks. So when you compare, you must have both similarities and differences. Now, there are many similarities. So both types of mutations, they are actually mutations. So mutation means spontaneous or random changes of genetic material such as DNA or chromosomes in the cells. So both will involve these random changes of genetic material such as DNA and chromosomes in the cells, right? So both will cause variation because they cause changes in the genetic material. So they'll cause changes in characteristics of the individuals. So they'll cause variation, which are differences in the characteristics in a species. Both cause genetic diseases, which may be serious. Both types of mutations can be passed on to or have effect on offsprings, on the children, right? If the mutations occur in the gametes. Okay, so the mutations can be passed down from the parent to the gametes, and the effects will be in the children or offsprings. Now, both types of mutations are can be caused by mutagens, which are substances that cause mutations, such as alpha rays and benzene. Right, differences. Gene mutation is on a smaller scale. The changes are on a smaller scale. Gene mutation involves changes in nucleotide base sequence. The nitrogenous bases are adenine, thymine, cytosine, and guanine. So maybe adenine in the sequence of bases is replaced by thymine or guanine. So that is a change in the nucleotide base sequence, okay, in the gene or in the RNA molecule inside the chromosome. Whereas chromosomal mutation involves changes on a larger scale in the structure of a chromosome, okay, or in the number of chromosomes. So more genes are involved, okay. Changes involve more genes for chromosomal mutation. Now, gene mutation is a small scale change in a single gene where maybe 1 or 2 bases or a few bases are different from the normal ones. But in chromosomal mutation, it's a large scale change in whole chromosomes, okay, where a chromosomal segment may be deleted or duplicated, or whole chromosome extra chromosome may be added into the cells of the gametes. Now, gene mutation, it is caused by base substitution, base deletion, and base insertion, where the base are the molecules of adenine, thymine, guanine, and cytosine. Now, we have discussed that in another video. So, chromosomal mutation is caused by deletion, duplication, inversion of whole chromosome segments, which are bigger, okay? And all these can cause changes to the structure of the chromosomes, whereas non-disjunction of homologous chromosomes in meiosis cause changes in number of chromosomes, okay? Now, examples of diseases caused by gene mutation are, think of the word CHATS, C-H-A-T-S, okay? Now, the C, what is the C for? Cystic fibrosis. H is for hemophilia. A is for albinism. T is for thalassemia. And S is for sickle cell anemia. So I just put 3 of them here, 3 diseases. Now, examples of diseases caused by chromosome mutation, you can give just one example like Down syndrome or Turner syndrome, Klinefelter syndrome, Cri-du-Chat syndrome, and so forth. Okay. So if in the exam you are asked to compare diseases caused by gene mutation, a disease caused by gene mutation with a disease caused by chromosomal mutation, okay, you can use some of these points here also, right? Let's say you're asked to compare albinism or hemophilia with Down syndrome or with Turner syndrome. So all these similarities and differences may apply, okay? Just adapt it to the specific disease. Now, here's a brief analysis of some questions that have been asked in the exam from Chapter 12, Variation from 5 syllabus. So, year 2021, discontinuous variation characteristics and causes of variation such as independent assortment of homologous chromosomes, okay? And then also comparison between Down syndrome and Cri-du-Chat syndrome. And in another year, year 2023, continuous and discontinuous variation in twins were asked, okay? So if you want to try the SPM questions, please do buy the book, Past Year Question Book, and try, okay? Try the questions from 2021 and 2023. Now, what is my ramalan or forecast questions for future exams like this year's SPM and so forth? Okay, this is my take on it. So, karyotypes may be asked, where you have to identify what genetic disease is involved, and then you may be asked to compare different types of gene mutation or chromosomal mutation and the characteristics of these types of mutation. So a comparison of genetic diseases is quite possible, okay, diseases caused by gene mutation or chromosomal mutation. So when making a comparison of genetic diseases where there are changes in the genetic material in the organism, in the cell, we can compare based on a few things. These are my suggestions. First, give the name of the disease, okay, maybe based on karyotype or on some symptoms. And then the patient's gender. Is it only males are involved or females are involved, or it could be either one, right? It can be either one. So what is the type of mutation involved? Is it gene mutation? Is it deletion? Chromosome mutation such as deletion or non-disjunction and all that? What's the type and the cause of the mutation? Then the number of chromosomes in each diploid cell for the particular patient that suffers from the genetic disease. The symptoms and characteristics can be compared, okay, between different diseases like hemophilia, compare with Down syndrome, right? Now, effects also, you can compare the effects of the 2 different genetic diseases. And remember to state the similarities too. For example, both hemophilia and Down syndrome cause variation, which are the differences between organisms of the same species. And both these diseases involve changes to the genetic material in the cell. All right? So you can refer to the upcoming shorts video, which I'll put up in 1 or 2 days, on how to compare 2 genetic diseases. And this is the strategy you can apply to all the different types of genetic diseases. Okay, so it will help you to be able to compare well the two genetic diseases. Okay, let's start. Chromosomal mutation. Definition. So mutation is a spontaneous and random change of genetic material or DNA or chromosomes in an organism cell. All right, think of genetic material, then in brackets DNA and chromosomes. All right, these all contain genetic material in an organism cells. All right, so how is it caused? When a person is exposed to mutagens or substances that can cause mutation, such as ultraviolet rays, ionizing radiation, benzene, food preservatives, and so forth. Okay, so effect. Chromosomal or gene mutation can change the characteristics of an organism. All right, can be asked what is the effect? The effect of this chromosomal or gene mutation that we have discussed in previous video can change the characteristics or the phenotype of an organism and lead to variation. Okay, so types of chromosomal mutation. A changes to the chromosomal structure and B changes to chromosomal number. So I've enlarged the diagram for the concept map for chromosomal mutation. This is a very good diagram, all right, not in the textbook. So you can take note of this and memorize the chart here, right? So chromosomal mutation consists of two types. The first is when there's a change in the number of chromosomes, and the second when there's change in structure of chromosomes. Okay, now we will start off discussing the change in structure of chromosomes, and these are due to the processes, abnormal processes like deletion, which causes Cri-du-Chat syndrome, where the cry of the baby is like a meowing kitten, and then inversion, duplication, and translocation. All this occurs to a chromosome segment that is relatively large compared to just mutation, where there is a change in nucleotide sequence. Okay, so for example, as we can see here, if the segment Q is taken out and removed, okay, and then the two ends join together, then you get a shorter chromosome. Now, this is called deletion, right? The process of deletion, which is one type of mutation, right, that changes the structure of the chromosomes. Then, secondly, we'll discuss change in number of chromosomes, which causes Down syndrome, where there are 47 chromosomes per diploid cell, Turner syndrome, where there are 45 chromosomes per diploid cell, Klinefelter syndrome, and Jacob syndrome. Okay, so the acronym for all these different diseases that are caused by chromosomal mutation is King, Jacob, Turn down, Chris. King for Klinefelter, Jacob for Jacob syndrome, Turn for Turner syndrome, Down for Down syndrome, and Chris for Cri-du-Chat syndrome. Okay, the only odd man out is Cri-du-Chat syndrome, which does not have a change in, which do not show a change in chromosomal number. Okay, it has the child will have 46 chromosomes, just like the chromosome number 5 is different because part of its arm has been deleted. So remember, King, Jacob, Turn down, Chris. So first we're going to discuss the first type of chromosomal mutation, which causes changes in the chromosomal structure of a particular chromosome. All right. So the changes in the chromosomal structure is sometimes called chromosomal aberration. So there are four types of chromosomal aberration that we are going to study. D-I-D-T, DT, okay, or ditten. So first is deletion, where a segment of the chromosome is lost, like the segment Q, is removed, is cut off, and then is removed. All right. The second type is called inversion, where a segment of chromosome is inverted or reversed or rotated in 180 degrees, right? So instead of the PQRST, now is PT, okay? The segment from behind here has been joined to the P, and now Q is joined to U. So this whole segment has been reversed or rotated 180 degrees, okay? Now the third type of chromosomal aberration that causes changes in the chromosomal structure is duplication, where a segment of the chromosome is repeated and then it joins to the original chromosome. Okay, like two segments of Q. Okay, so the Q is duplicated. the Q segment. Now, lastly is translocation, where a segment of chromosome from another chromosome that is of a different type, a non-homologous chromosome, joins to the first chromosome here, all right? And that means it has changed location. Translocation means change the location of the segment of chromosome, okay? So let's discuss further. Now, so changes in chromosomal structure causes changes to the gene sequence in a chromosome, and the chromosomal aberration means the structural changes that cause abnormality to the chromosome. Okay, so actually chromosomal aberration means a type of chromosomal mutation where there is change in the structure of the chromosome. Okay, so the four types of chromosomal aberration are deletion, duplication, inversion, and translocation. DT. Okay, DT. So there are four types of chromosomal aberration which causes changes in chromosomal structure. So the chromosomal aberration are the different types of chromosomal mutation which causes changes in chromosomal structure. The first type of chromosomal aberration or mutation is deletion. A part or segment of the chromosome is deleted or lost, for example, segment Q, and this causes a loss of a few genes, right? So let's say this is the normal chromosome. So a segment of the chromosome will break off and then it is deleted or removed entirely from the original chromosome, all right? And then the two ends here join back together. So now the chromosome is shorter and one segment is lost, okay, which causes a loss of a few genes. Or if we were to write it in this format, PQRSTU, the Q is lost, segment Q is lost, so it becomes PRSTU. So this type of chromosomal aberration or mutation is called deletion. And one example of a disease caused by deletion is Cri-du-Chat syndrome, the cry of the cat syndrome. So duplication is the second type of chromosomal aberration or mutation where there's a change in chromosomal structure. A part of a chromosome is copied and repeated, let's say segment Q, which causes repetition of the gene sequence, right? So this is the original chromosome, PQRSTU. Now, the Q segment is copied and repeated and then is joined to the chromosome, original chromosome. So the chromosome becomes longer with an extra few genes here, okay, for the Q segment, in the Q segment. So PQRSTU becomes PQQRSTU. Okay, this is duplication. Then thirdly, the third type of chromosomal aberration is inversion. So a segment of chromosome is reversed end to end at 180 degrees and then rejoined. This causes change in the DNA sequence in the chromosome. Okay, for example, if you look at this picture, so the original chromosome has these segments, PQRSTU, but the segment breaks off at Q and at T, and then it inverts and it is reversed. The order is reversed. Okay, so there's sort of some rotation going on here. So now the T is joined to the P and the Q is joined to the U. So the rotation is at 180 degrees, okay, from this part to this part. So now this is a mutant chromosome because the sequence of genes and the sequence of the nucleotide bases of a large part of nucleotide bases has been inverted. All right. So PQRSTU becomes P joined to TSRQ. This is the inverted section or segment, then U. Okay. So this is the process of inversion of a segment of chromosome. Okay. Inverted and rotated 180 degrees. Now, next, the last one is translocation. Trans means to change. Location means the place, the position. Okay. So a part of a chromosome is cut off, it breaks off, and it joins to another non-homologous chromosome. That means another chromosome that is of a different type, carrying different genes, right? So two chromosomes are involved here in this case for translocation. First, we have chromosome number 1 with the sequence of segments PQRSTU, right? And then another chromosome ABCDEF. So possibly, let's say the segment breaks off at T, okay, for chromosome number 1, and the segment breaks off at D here, or at E here, for chromosome number 2. Then they exchange places. Okay, each segment joins to the other chromosome. Okay, so the segment T and U will join to this chromosome number 2. From chromosome number 1, segment T and U joins to chromosome number 2. Okay, it breaks off and joins to chromosome number 2. Whereas the segment E and F from chromosome number 2 joins to chromosome number 1. So finally, we'll get this result. PQRS, then after the S segment is followed by the E and F segment from chromosome number 2. Then for chromosome number 2, A, B, C, D segments, and then the segments from chromosome number 1, T and U joins to it. So now these two chromosomes are called mutant chromosomes. That means there is mutation already in these two chromosomes, right? Due to the process of translocation, exchanging locations. Okay, the two segments exchange their location, the place where they are attached to. So chromosome number 1, PQRS-TU becomes PQRS. TU has broken off, then EF, these segments join to the first chromosome. Okay, so PQRS-EF, PQRS-EF. Then for chromosome number 2, ABCDEF becomes ABCD-TU. Now, so we finished discussing changes in chromosomal structure with the acronym DIDT. Now, for the second type of chromosomal mutation, it involves changes in chromosomal number. All right. So many of these types of chromosomal mutation are caused by a process called nondisjunction, which we'll discuss afterwards. Okay, which is the failure of homologous chromosomes to separate in meiosis, in Anaphase 1 or Anaphase 2 of meiosis. So examples of diseases caused by changes in chromosomal number are KLINEFELTER syndrome, where there are 3 sex chromosomes in each diploid cell, XXY. Jacob syndrome, where there are also 3 sex chromosomes, but it's XYY in each diploid cell. Turner syndrome, where there is only 1 sex chromosome in each diploid cell, that's the X chromosome. And for Down syndrome, there are 3 chromosome number 21 in each diploid cell of the child. Okay, so let's find out more about these interesting types of genetic diseases. Now, the second type of chromosome mutation is occurs where there are changes in chromosomal number, okay, instead of structure. Chromosomal number changes. So a change in chromosomal number causes a diploid organism to lose 1 or more chromosomes, as in the case of Turner syndrome patient who has 45 chromosomes in each diploid cell instead of 46 chromosomes, because 1 chromosome is lost. Okay, or the diploid organism may gain 1 or more chromosomes, as in the Down syndrome child who has 47 chromosomes in each diploid cell instead of 46 chromosomes, as in a normal cell, right? So what is the cause of the change in chromosomal number? The change in chromosomal number occurs due to a process known as nondisjunction during meiosis in the formation of gametes. So in the formation of gametes, there is already an abnormal process. Okay, and then when the abnormal gamete is produced, if it undergoes fertilization, then it will produce a child with a genetic disease where there's a change in chromosomal number. So non-disjunction, what does it mean? The word disjunction means separation. Non-disjunction means non-separation. All right. So non-disjunction is a process or phenomena in which homologous chromosomes fail to separate during Anaphase 1 in meiosis. That means non-separation of homologous chromosomes during Anaphase 1. Or sister chromatids fail to separate during Anaphase 2 in meiosis 2. Okay. So we know that in meiosis 1, in Anaphase 1, homologous chromosomes should separate. Okay, let's look at this now. Homologous chromosomes should separate and be pulled to opposite poles by the spindle fibers, right? But let's say in this case, these 2 long chromosomes, they are homologous chromosomes of the same types. These 2 copies are pulled to only 1 pole. They do not separate. Now, how can this occur? Now, the reason is spindle fibers may fail to form normally in meiosis. For example, on the right-hand side here, the spindle fiber may not be completely formed or well formed. So the spindle fibers on the other side will pull the 2 chromosomes over, okay, to the same pole, right? So when spindle fibers fail to form, normally in meiosis, there's no separation of the homologous chromosomes or sister chromatids in Anaphase 2. Okay, so sister chromatids should separate in Anaphase 2 in meiosis because they are pulled by spindle fibers to opposite poles. But if there is a mutation or because of exposure to certain substances that cause mutation, then maybe the two sister chromatids fail to separate because spindle fibers fail to form to pull them to opposite poles. This may occur in Anaphase 2. Okay, so whichever situation it is, A or B, okay, A involves homologous chromosomes that fail to separate, B involves sister chromatids that fail to separate. These two cases are called non-disjunction. Okay, now whichever case of non-disjunction, when there's no separation of homologous chromosomes or sister chromatids in meiosis, then it will produce abnormal gametes. The parent will produce abnormal gametes with abnormal number of autosomes, such as a gamete with 2 chromosome number 21 instead of 1 chromosome number 21. Okay, or abnormal number of sex chromosomes. If the sex chromosomes undergo non-disjunction, then it will cause the gametes to have abnormal number of sex chromosomes, as we'll see afterwards in the diagram. So the effect of non-disjunction is that it causes abnormality in the number of chromosomes in a gamete because the homologous chromosomes fail to separate or the sister chromatids fail to separate. Okay, so the gamete formed at the end of meiosis may lose one or more chromosomes, such as a sperm with no sex chromosomes, or gain one or more chromosomes, such as a sperm with 2 sex chromosomes, X and Y. Now the normal sperm should have either an X chromosome or a Y chromosome. Okay, but abnormal sperm formed through non-disjunction may have 2 sex chromosomes, X and Y together, okay, in the sperm. Ovum with XX chromosomes instead of just 1 X chromosome, ovum or secondary oocyte. Now, another abnormal situation is when the sperm or ovum has 2 chromosomes number 21 instead of 1 chromosome 21. So this picture is also in your textbook, all right? So let me explain it. It shows 2 examples of non-disjunction. One that occurs during Anaphase 1, which is A, and in B, non-disjunction that occurs during Anaphase 2, in meiosis 2. Okay, so let's talk about non-disjunction during Anaphase 1 in meiosis 1. Now this is the diploid cell and there are 4 chromosomes inside. So 2 copies of the short chromosomes and 2 copies of the long chromosomes. Okay, so if the abnormality, which is non-disjunction, occurs during meiosis 1, that means it causes the homologous chromosomes not to be separated. Homologous chromosomes fail to separate in meiosis 1, right? In Anaphase 1 of meiosis 1. Now, the top short chromosome at the top here, the 2 short chromosomes, they separate in the normal way, okay? 1 chromosome to each pole, being pulled by the spindle fibers. But what happens if the spindle fibers do not form, okay, in this part of the cell? So the spindle fibers on this side, on the left-hand side, pull the 2 homologous chromosomes to the same pole, okay? So abnormality is non-disjunction or non-separation of the homologous chromosome pair here to in Anaphase 1, and it causes both chromosomes to go to one pole, okay? Now, then what happens in meiosis 2 to this cell? So the cell will divide into 2, and this is the intermediate daughter cells formed. So in the first daughter cell on the left here, we have 1 short chromosome and 2 long chromosomes, right? Now they are colored red and blue to differentiate between the paternal and maternal chromosomes, okay, of the same type. So this is an abnormal cell, and this cell also is abnormal because it has only 1 short chromosome. It doesn't have any long chromosomes. By right, a normal cell at this stage will have 1 short chromosome and 1 long chromosome, okay? So these 2 are abnormal cells because this cell also has 2 long chromosomes, so it's not normal. Now, after meiosis 2, now at meiosis 2, in Anaphase 2, sister chromatids separate. So these 3 chromosomes here will be lined up in a single file, and then the sister chromatids will separate, okay, in this diagram, the opposite poles. So the separation of the sister chromatids is normal. So it's called normal disjunction or separation of sister chromatids during Anaphase 2, right? But the cell is really abnormal. And in this cell with one short chromosome, the sister chromatids of the short chromosome separate in Anaphase 2 of meiosis 2. Now then, what do the resulting daughter cells look like? Okay, what do the daughter cells look like? So imagine that cytokinesis occurs and the cell will divide at this in the middle here, right? So the first daughter cell, which is a gamete form, will have 3 chromosomes, one short chromosome. This is a normal case, but it has 2 long chromosomes. It should only have 1 long chromosome. Okay, so that means it has an extra long chromosome, so it's N plus 1, haploid number plus 1 extra chromosome. Same with this second cell, right, on this side. Now, the third cell here is a product of this intermediate cell, so only 1 short chromosome will be found in this gamete, and there's no long chromosome. So it's missing 1 long chromosome, so it's N minus 1, okay? Because each sister chromatid here will become a new chromosome, right? But here, there's no sister chromatid of the long chromosome separating, so there's a missing long chromosome. Now let's look at the other case in which the non-disjunction occurs during Anaphase 2 in meiosis 2. So at first, in Anaphase 1, there is normal disjunction or normal separation of homologous chromosomes to the opposite poles. Okay, so the spindle fibers will pull the chromosomes apart, the homologous chromosomes apart, and then they move to opposite poles, and the resulting daughter cells, intermediate daughter cells, will be like this. Each intermediate daughter cell is normal. It has 1 short chromosome and 1 long chromosome. Okay, same for this one. But then what happens if an abnormality or mutation occurs in the Anaphase 2? There's non-disjunction of sister chromatids during Anaphase 2. Okay, so let's look at this. Now we talk about the normal condition first. Now let's say this cell undergoes the normal process. In Anaphase 2, sister chromatids separate and move to opposite poles, right? For the short chromosome, and this happens to the short chromosome and then also to the long chromosome. So this will produce gametes that are absolutely normal with 1 short chromosome and 1 long chromosome in each cell. Okay, when this cell divides into 2 through cytokinesis, right? So this is the haploid number of chromosomes, where there's 1 copy of each type of chromosome in the cell. But let's look at the second case where the abnormality occurs and there's non-disjunction in sister chromatids or non-disjunction of sister chromatids during Anaphase 2, right? So the top chromosome here, the short one, separates normally. The sister chromatids separate normally. Sister chromatids move to opposite poles of the cell when the spindle fibers contract and pull them apart. Let's say there's an abnormality here or mutation where there's non-disjunction, non-separation of sister chromatids for this chromosome, this long chromosome. So both the long chromosomes move to the same pole, okay, still attached at the centromere, right? Now, after Anaphase 2 is over, the cell divides into 2, and the result is like this. All right, now you can see here, in the first cell on the left here, there'll be 1 short chromosome, and then these 2 sister chromatids that are still attached with each other, they will separate. They will separate and each will form a whole new chromosome. So there'll be 2 whole new chromosomes in this cell. Okay, so this is N plus 1 because there's an extra copy of the long chromosome. This is an abnormal cell. On the other side, there's a missing long chromosome, so only 1 short chromosome. This condition is N minus 1, the haploid number minus 1. So here we can see that non-disjunction that occurs in Anaphase 1 or Anaphase 2, okay, can cause the abnormal gametes to be formed with abnormal number of chromosomes, such as these 3 cases. Only 1 case here is normal. Okay, gametes are of the normal chromosomal number. So let's discuss this diagram, which is also in your textbook. Actually, it's quite easy to understand. This picture shows you 2 possibilities. One is when non-disjunction occurs in a female, okay, the non-disjunction of sex chromosomes XX occurs in a female during oogenesis. And the second one is if there's non-disjunction of sex chromosomes XY in a male during spermatogenesis or formation of sperms. So let's take the first case. Oogenesis is the formation of the secondary oocyte and the ovum. So what happens if there's non-disjunction of sex chromosomes XX during Anaphase 1? Okay, so here we see only two pairs of homologous chromosomes. Assume there are more pairs, but we just show two pairs. Okay, so the top pair, the short chromosomes here, they separate normally. All right, the one of the homologous chromosomes goes to one pole and the other one goes to the opposite pole. Okay, so this is normal disjunction, normal separation of homologous chromosomes in Anaphase 1. But what if this pair here, which are the sex chromosomes, undergo non-disjunction or non-separation? The two homologous chromosomes XX fail to separate and they move to the same pole in Anaphase 1. Okay, so this will give rise or produce abnormal secondary oocytes after that. Okay, so this first secondary oocyte has zero sex chromosomes because none of the sex chromosomes XX move to this left pole, right? So no sex chromosomes, but other autosomes, other chromosomes like autosomes are present, right? Then whereas for this other side here, the right hand side, you will give rise to an ovum with two X chromosomes. So by right, ovum should have only one X chromosome if there is normal disjunction or separation of homologous chromosomes. But in this case, both the X chromosomes move to the same pole, so it produces an ovum with extra one sex chromosome. Okay, so XX. Now what happens if the abnormal secondary oocyte is fertilized by a normal sperm? So normal sperms are like this. A normal sperm can have one sex chromosome X or one sex chromosome Y. That is a normal sperm. Okay, so if the normal sperm, we call it the X sperm, fuses with the abnormal ovum that has zero sex chromosomes, then the sex chromosome X and O will, this one none, no sex chromosome, will come together. So it will be a zygote that has only one X chromosome. Okay, so finally the zygote will develop into a child, and all the diploid cells of the child has just one X chromosome. So this gives rise to Turner syndrome in the female. It's a female with Turner syndrome. Now the second case is if this abnormal ovum fuses with the Y chromosome, okay, Y sperm, with the Y sperm, okay, then the zygote will have only one Y chromosome in its cell. Now this zygote cannot survive because the Y chromosome is very small and it has fewer genes than the X chromosomes. Okay, so this zygote will die. Now let's take the second case where the ovum has two X chromosomes. This is an abnormal ovum. If it fuses with the X sperm, then the three X chromosomes will come together in the zygote and produce an individual or female with triple X syndrome, where every diploid cell has three sex chromosomes X. So we don't discuss this type of condition in this textbook, so we will not discuss it. Now let's look at the second case. If the abnormal secondary oocyte with two X chromosomes fuses with Y, the Y sperm, then it will produce a child with two X chromosomes and one Y chromosome in each diploid cell. So this is a male with KLINEFELTER syndrome. Okay, male because of the Y chromosome, and then he suffers from KLINEFELTER syndrome where there is an extra X chromosome. Okay, so let's go on to the next case where non-disjunction of sex chromosomes XY occur during spermatogenesis in the male. Okay, so again we come to this diploid cell here, and now the sex chromosomes are XY. They fail to separate from each other. They move to the pole on the right. Okay, in the formation of sperms. So finally, the sperm form, one half of the sperms form will, 50% of the sperm form will have no sex chromosomes because the two sex chromosomes move to the right pole. Okay, so the left pole has no sex chromosomes. So this produces a sperm without the sex chromosomes. And the other sperm has two sex chromosomes XY instead because of non-disjunction of the sex chromosomes in meiosis 1 during Anaphase 1. Now, what happens if a normal secondary oocyte is fertilized by these abnormal sperms? Okay, so in the first case, this normal secondary oocyte fuses with this sperm without sex chromosomes. So it produces a Turner syndrome child with one X chromosome in each diploid cell. So similar to the first case here. However, the way that it's produced is different. Okay, in this first case, it's the ovum that has no sex chromosomes that is abnormal. In this second case, it's the sperm that is abnormal without any sex chromosomes. Okay, but still the result is the same, that is Turner syndrome child is born. Now for the second case, for the sperm that has two sex chromosomes, X and Y, if it fuses with a normal secondary oocyte, it will produce a child with XXY chromosomes. All right. So again, this is Klinefelter syndrome child who is a male, similar to the case here, except that in this first case, all right, it's the ovum that's abnormal with two X chromosomes, right? In the second case here, it's the sperm that's abnormal with two sex chromosomes, X and Y. So here we find out that non-disjunction of sex chromosomes can occur during the formation of gametes, giving rise to if the gametes are then fertilized. Okay, after non-disjunction, if there's fertilization, then only will produce the child with the abnormal number of chromosomes, such as Turner syndrome and Klinefelter syndrome. Now, how about Jacob's syndrome? How is Jacob's syndrome child produced? Now, for Jacob's syndrome, is the non-disjunction in meiosis 2 for Y chromosome. The Y chromosome fails to the chromatids of the Y chromosome fail to separate from each other. Okay, so the two sister chromatids fail to separate, so both move to the same pole, and each sister chromatid becomes one new chromosome. So it becomes Y1, two Y chromosomes in the abnormal sperm. Alright? Whereas the other sperm here has 0 sex chromosomes. So X, the ovum is normal, so a normal secondary oocyte is fertilized by the abnormal sperm. So in the first case, X plus O becomes XO. So this is again Turner syndrome child that's formed with only one sex chromosome in each diploid cell. Okay, one X chromosome. Now, this is the one. If the abnormal sperm with two Y chromosomes fuses with a normal secondary oocyte with one X chromosome, okay, in the process of fertilization, then the resulting zygote will have one X chromosome from the ovum and two Y chromosomes from the sperm. Okay, so XYY. So the child with the 3 chromosomes in each diploid cell, XYY chromosomes, okay, has a Jacob syndrome. It's a male, a sterile male that cannot bear children. So let's dive deeper into the diseases caused by chromosome mutation. All right, especially we're going to talk about the 5 types of diseases and discuss their symptoms in more detail, and then look at their karyotypes. The karyotypes are photographs of the homologous chromosome pairs in each cell, okay, each diploid cell. So our memory tip or acronym is King, Jacob, turn down, Chris. Okay, remember, you just need to repeat this many times and then relate it to the 5 different diseases that we're going to discuss. Okay, King, Jacob, turn down, Chris. Okay, King for Klinefelter syndrome, J for Jacob syndrome, T for Turner syndrome, turn down for turn for Turner syndrome, down for Down syndrome, and Chris for Cri-du-Chat syndrome. Now, how to remember Cri-du-Chat syndrome? Look at the word Cri. If you place the letters closer together and then you just rotate it anti-clockwise, 90 degrees, okay, you will come to this structure or symbol. So it looks like the number 5. Okay, so this one, the Cre itself looks like a 5 that is lying down. All right, so think of chromosome number 5. Chromosome number 5 is involved. Actually, Cre-du-Chat syndrome is caused by a deletion of a part of chromosome number 5, where a part of chromosome number 5 has been removed. All right, so the D here stands for deletion. All right, remember that. Cre stands for 5 and D for deletion. Deletion of a part of chromosome number 5. And this causes changes to the structure of the chromosome, right? And then the C, the two C's here, think of cry like a cat. Cry like a cat's meow, okay, or cry like a cat's cry. So the child will cry in a way that sounds like a kitten meowing away, all right? Okay, so let's look at other points now. The chromosomal number on the whole is normal. It's 46 chromosomes in each of the diploid cells of the child. But it's the structure of chromosome number 5 that is different from the normal structure because a part of it has been deleted, right? So 2n equals to 46 chromosomes. This is the diploid number for this child, or 44 plus XY for males, or 44 plus XX for females in each diploid cell. The baby can be male or female. It's just that the chromosome number 5, which is an autosome, not involved in sex determination, right? Chromosome number 5 is an autosome, not involved in sex determination. It has a part of it deleted, right? So the symptom is crying like the cry is like a cat's cry or a cat meow. Right, so now we look at the karyotype of Cre-du-Chat syndrome. Everything is normal, okay? There are 46 chromosomes, just that for chromosome number 5, it's shorter than normal because there's a deletion on the short arm of chromosome number 5. Okay, the upper part is missing. So that deletes or removes some genes from the karyotype. So this causes the baby to have abnormal characteristics. The cry of affected infants is like a meowing kitten or cat. Secondly, the patient will show delayed mental and physical development. Okay, so it shows delayed development. They may walk later, talk later, and so forth. So there is mental and physical retardation, right? So they cannot develop fully and they develop very slowly. Most patients die during childhood. So if they manage to grow up, you can see that the face will be white, have white space eyes and small jaw. Now this is an extra feature, it's not in the textbook, all right? But the important part here, all these characteristics are listed in the textbook and you have to know all these characteristics, okay? Now how to remember Down syndrome, the facts about Down syndrome, important facts. So here's a creative method. Look at the word D, just underline the word D and you will see a letter forming, which is number 2. Okay, ignore the vertical section here. So imagine it's a 2 and then plus the O, which stands for 1. So 21. Okay, it's like 21. So there's one extra chromosome 21 in each cell. Okay, one extra chromosome 21 in each of the diploid cells. So instead of 2 chromosomes 21, there are 3 chromosomes number 21. Okay, in the Down syndrome child. Next, we have discussed the significance of D and O. Now let's look at this next one, W. W looks like an inverted tree, right? Like a tree that's lying down, all right? So 3 chromosomes number 21 are involved, right? In each diploid cell. So the child, Down syndrome child, has 3 chromosomes number 21 in each diploid cell. Okay, so W stands for 3. Now next, the N. N, think of nose, all right? So the symptom is a flat nose. Okay, Down syndrome child, their nose at the bridge here is quite flat, right? Now let's go back to the chromosomal number. So chromosome number will be 2n plus 1 because of the extra chromosome 21. So it becomes 47 chromosomes in each diploid cell instead of 46, which is the normal number. And if you divide up into the autosomes and sex chromosomes, it's either 45 plus XY or 44 45, all 45 autosomes plus XY or 45 plus XX. Now why 45? Because of the extra chromosome 21. Okay, instead of 2 chromosomes 21, the child has 3 chromosomes 21. So a common question asked is, how is Down syndrome child actually formed? Right? So this is a common question in SPM. Now, any abnormality during gamete formation in meiosis can cause genetic diseases such as Down syndrome. So let's see how to explain the cause of Down syndrome. Two processes are involved: non-disjunction or non-separation of homologous chromosomes or sister chromatids in meiosis, and then fertilization of the abnormal gamete. Okay, so non-disjunction and fertilization. So firstly, spindle fibers may fail to form during Anaphase 1 or Anaphase 2 of meiosis during formation of gametes. Thus, the homologous chromosomes number 21 may fail to separate in Anaphase 1. All right. So if homologous chromosomes number 21 fail to separate in Anaphase 1 of meiosis, then this process is actually called non-disjunction of the chromosomes number 21, homologous chromosomes number 21. It's not the sex chromosomes that undergo non-disjunction. Now this is only for for us to take note. No need to write down in the explanation. That is not the sex chromosomes. Next, abnormal gamete will be formed because of the non-disjunction, and this abnormal gamete has 2 chromosomes number 21 that fail to separate from each other. So if fertilization between a normal gamete with 23 chromosomes, now this may be a sperm, all right, normal gamete, and maybe it's the mother's ovum or secondary oocyte that is abnormal. So if the normal sperm fuses with an abnormal ovum, okay, normal gamete fuses with abnormal gamete that has 24 chromosomes or 2 chromosome number 21, then resulting zygote will contain 47 chromosomes, okay, 23 plus 24, and normal gamete will provide 1 chromosome 21, and the abnormal gamete provides 2 chromosomes 21. So total will be 3 chromosome number 21, and this is how the Down syndrome child is formed. So let's look at the karyotype for Down syndrome child. Trisomy 21, 3 chromosomes 21. So there are 2 copies of each type of chromosomes except for chromosome 21. There are 3 copies of chromosome 21. So circle this part here to note the abnormality. Okay, is with the chromosomes 21. So chromosomal number, as we have said, is 2n plus 1 equals to 47 chromosomes in each diploid cell. The child has, or the person has 3 chromosomes number 21. Okay, or 3 number 1, 21 chromosomes. Better write 3 chromosomes 21. 3 chromosomes 21. Okay, 3 chromosomes number 21. Now 45 plus XX or 45 plus XY, depending on whether the child or the person is male or female. So what are the characteristics of a Down syndrome patient? So remember at least 3 characteristics. If you can remember all, it's better. If you can remember all, okay, all the 5. Now these are all taken from the textbook, so we follow the textbook. Slanted eyes. The eyes are slanted upwards at the ends here. Okay, flat nose. So the nose is flat. The bridge of the nose is usually flatter than normal, and the protruding tongue, right? So the tongue is a bit longer than normal, so it protrudes out from the mouth. Broad forehead. Forehead is broad, and mentally retarded. Another characteristic is stunted growth also. Down syndrome patient tends to be a bit shorter. Okay. So can this characteristic be passed on to the next generation? Normally, it's not passed on. It's not inherited normally. Okay. Now, how to remember KLINEFELTER syndrome? So this is another disease that is caused by a change in chromosomal number. Okay. So where there are 3 chromosomes, 3 sex chromosomes instead of 2, with an extra X chromosome, XXY, okay, instead of XY. So how to remember? Now you look at the letter K. Okay, we start with the name itself. The letter K, if you twist it a bit, it looks like an X, right? second X. So that means the abnormality is that there's one extra, think of X, extra, there's one extra X chromosome, or extra one X chromosome. The person has an extra X chromosome, okay, extra X chromosome. So then the K can also stand for king. Klein sounds like king, right? So think of king. A king is a male. So this patient is a male. Why? Because he has the normal X and Y chromosomes, except that there's an extra X chromosome. Okay? So he has XY chromosomes. As long as the patient has an X chromosome, he is considered a male, okay, with characteristics similar to a male. Okay, some characteristics are similar to a male. So the chromosomal number is 2n plus 1 because of the extra X chromosome, you have plus 1 more. Okay, so 47 chromosomes. And if written out, you break it down into autosome and sex chromosomes, is 44 plus XXY, 3 sex chromosomes. So it's a male because of the XY chromosomes, but there are certain characteristics that are female, okay, like for female because of the extra X chromosome. Okay, so symptom, think of the second letter. Look at the second letter. It's an L. Long legs and hands. That's what textbook writes. Some books write long legs and arms. The whole arm is longer than for a normal person. And the person tends to be taller in stature, okay, than normal people. So let's look at the karyotype for Klinefelter syndrome. Now, all the autosomes will have 2 copies for each type. There are 2 copies for each type of autosome, right? So autosomes are normal, 44 autosomes in the diploid cell. It's the sex chromosome that has abnormality. So instead of one X and one Y chromosome, there's an extra X chromosome here. Okay, so we circle this part. This is where the abnormality is. 2 sex chromosomes X. So chromosomal number is 2n plus 1 equals to 47 chromosomes with an extra X chromosome. So if you break it down, it's 44 plus XXY. Okay, characteristics, again, you must base it on the textbook characteristics. Sterile man. Sterile means that they cannot produce children. They cannot produce sperms, right? So the patient will have small testes that fail to produce sperm. So that's why they are sterile. They cannot have children, bear children. Okay, even if the wife is not sterile, if the wife is fertile, the man is sterile, then cannot bear children. Now, that's because of the failure to produce sperms. The voice and chest similar to a woman, so there may be slight development of the breast, and the voice may be a bit high pitched. Okay, has long legs and hands or long legs and arms, and reduced facial hair. These are extra points, but remember the characteristics here, the 4 characteristics. Think of a man but with some women's characteristics. All right. Now this condition is not that common. Okay, it's considered rare. So number 4, Jacob syndrome. How to remember? So look at the J. It has some. When we write the letter J, there is a horizontal cap on top. Okay. So think of it as being pressed down a bit. Then it forms a V-shaped structure. So it looks like a Y. So it's the Y chromosome that is different. There are 2 Y chromosomes. There's an extra Y chromosome in each of the diploid cells for a child with Jacob syndrome. Okay, instead of just XY, the child has XYY chromosomes. Okay, so look at the letter J and think of it being depressed in the center here, then you get the Y. Okay, but some fonts in our computer, they don't have the letter, the lead on top, the horizontal part on top. Now Jacob is the name of a male, right? So it's a male. Okay, Jacob syndrome child is a male, and he has X and Y chromosomes, right? So think of Jacob, male, so he must have X and Y chromosomes, but with an extra Y chromosome. So chromosomal number is 2n plus 1 equals to 47 chromosomes, extra Y chromosome. Or another way of writing it is we separate the autosomes and the sex chromosomes. So it's 44 autosomes plus XYY chromosome. Okay, so differentiate from KLINEFELTER syndrome. In KLINEFELTER syndrome, the sex chromosome is XXY. For Jacob syndrome, is XYY, one extra Y chromosome, right? So the symptom is, now what's the characteristics of symptom? Look at the letter A here. Okay, after J, look at A. A for acquire speech slowly. Another A is for ability. Just reverse it to disability. So this child has a learning disability in that he will learn things more slowly than a normal person. Okay, and actually another A is for acne. ACNE, acne. Acne means pimples. Textbook use the word pimples. So this child has a problem with acne on his face. Maybe it's a constant problem. Okay, always having acne or pimples on the face. How about the karyotype? So in the karyotype, there are 44 normal autosomes, and the abnormality is that the child has an extra Y chromosome, two Y chromosomes, one X chromosome and two Y chromosomes, right? So with the extra Y chromosome, extra genes, that means this causes abnormalities and symptoms as follows. The male is taller than normal, has problem with pimples on his face like this. So these pimples, in some books, the medical term is acne. Okay, so in the A for Jacob, the A can also stand for acne, but you have to write in brackets pimples because the textbook use the word pimples. The more widely used word is acne. All right, acne. So has problem with pimples on his face, slow in acquiring speech, as we say, acquire speech slowly, has learning disability, so learn very slowly, and has weak muscles. So this condition is called hypotonia. Now, lastly, Turner syndrome. Okay, the last of the syndromes that we are studying. How to remember? Now, for Turner syndrome, we do nothing with the T here. Just leave the T alone. Think of the word Turner. Or look at U. Look at the U. Now, first of all, Turner sounds like loner. Turner sounds like loner. And the U, think of Uno. Uno also sounds a bit like Turner. So Turner, the U for Uno. Uno means what? One. There's a card game called Uno. Okay, so it's one. In I think Spanish, Uno means one. So this patient has one chromosome Y, chromosome X. All right, cannot be one chromosome Y, because if a patient has only one chromosome Y, the patient cannot survive. Okay, they must have at least the X chromosome. So Turner is linked to loner, that means one lone chromosome X. Okay, so these two words, Uno and loner, help us to remember that there's only one chromosome X, a lone chromosome X, sex chromosome X. And then when you think of Turner, think of Tina, Tina Turner. So it's a girl. A child with Turner syndrome is considered a girl, right? Because of the lone X chromosome. There's no Y chromosome. If there's a Y chromosome somewhere, then it will be a boy or a male. So there's no Y chromosome. So the chromosomal number is 2n minus 1. Missing one sex chromosome X, right? Becomes 45 chromosomes, 44 normal autosomes, and the abnormality is that there is only one sex chromosome X. So X, O here stands for 0. Just one sex chromosome. Okay? So this is a sterile female. Sterile means cannot bear children, right? Because of the lack of another X chromosome. But because the child has only one X chromosome, so the symptom or characteristic is, again, we look at the U. This time for Turner, we look at the U. U for undeveloped breasts and ovaries. So the female secondary sexual characteristics are not so well developed, all right, because of the lack of another X chromosome. Normal female have two X chromosomes in each diploid cell. Okay, but the Turner syndrome child has only one X chromosome. So undeveloped breast and ovaries and undeveloped secondary characteristics for females. Now, this is the karyotype. So all the autosomes are normal. It's the sex chromosome again that is abnormal because there's only one X chromosome and no Y chromosome or no X chromosome. Okay, no other sex chromosome. So, as we said, chromosomal number is 2n minus 1 equals to 45 chromosomes. And break it down, the chromosomal number is 44 plus XO or X0. Now, one of the symptoms is the neck. The neck is web-like because of the extra skin folds here. So the neck looks a bit thicker than normal, all right, with the extra skin fold at the side. Characteristics. Follow the textbook. Sterile female, cannot bear children, has lack of secondary female characteristics, okay, such as undeveloped breasts and ovaries, and then web-like neck as shown here, and low IQ. Okay, low IQ. So those are, just now we discussed the 5 different syndromes, right? Cri-du-Chat syndrome, Down syndrome, and Klinefelter syndrome, Turner syndrome, and Jacob syndrome. Okay, so this is the summary of it, which you can look through. It's the same as what we discussed just now, but this is put in a table form and summary for you to read. All right. So here are some notes on the abnormalities in sex chromosomes caused by non-disjunction of sex chromosomes XX or XY. So we discussed all this just now. All right, you can just copy it or take note of it. So far we've discussed non-disjunction during meiosis, where homologous chromosomes fail to separate or sister chromatids fail to separate. How about mitosis? Does non-disjunction occur in mitosis? Yes. Non-disjunction can occur in mitosis, can be due to exposure to mutagens also or substances that cause mutation. So in non-disjunction, in mitosis, sister chromatids fail to separate and move to opposite poles. Okay, but there's a difference, right, between non-disjunction in mitosis and non-disjunction in meiosis. For mitosis, non-disjunction in mitosis produces somatic or body cells that are abnormal with abnormal chromosomal numbers. And this leads to diseases like cancer and the growth of tumors which contain abnormal cells with abnormal number of chromosomes, right? But the gametes are not affected because it's not the gametes that undergo non-disjunction during the formation of the gametes. Okay, it's the somatic body cells that undergo non-disjunction. So non-disjunction meiosis is different because it produces gametes with abnormal number of chromosomes during the formation of gametes. Now, if fertilization occurs involving the abnormal gametes, then the offspring will suffer from conditions like Down syndrome, Turner syndrome, Klinefelter syndrome, or Jacob syndrome, which are all genetic diseases. Okay, so these diseases occur due to non-disjunction during meiosis in the formation of gametes. So here's the last part, mutation of somatic cells and gametes. So mutation can occur in cells to produce variation in the population, and there are 2 types of cells in the body that can undergo mutation. Okay, the normal body cells are called the somatic cells that are diploid. For example, the skin cells, the cells of the organs like the liver, stomach, and so forth, right? And then the other type of cells are the haploid gametes that are found in the reproductive organs like testes and ovary. Okay, so what are the differences between mutation in somatic cells and gametes? This can be asked as an essay question. Okay, so or as a structured question. Now, mutation in somatic cells involves somatic cells such as skin cells, eye cells, and brain cells or nerve cells, and other types of cells like the white blood cells and so forth. So mutation in gametes, it involves germ cells that will produce the gametes, such as the secondary oocyte and the sperm. So it occurs in the reproductive organs. Now, mutation in somatic cells cannot be inherited by the next generation because the somatic cells are not used to form the zygotes of the next generation, the zygote for the next generation. Okay, yeah, in the body of the individual. That's all. Now, mutation in gametes can be inherited by the next generation if those gametes that are abnormal, that have mutation, are fertilized. Okay, if those abnormal gametes are fertilized. So disease is present only in the individual with the mutation in somatic cells. For example, a mutation in the skin cells due to exposure to strong ultraviolet rays, excess ultraviolet rays, can cause skin cancer, right? So that skin cancer is not passed down to the offspring, all right? Because it occurs in the somatic cells only, not in the gametes. Now, mutation in gametes, the disease is present in the individual, all right, with the mutation. That means maybe the gametes have the abnormal number of chromosomes, and then if the gametes are fertilized, then the effects of that mutation will be inherited by the descendants or by the offspring or children. Okay, can be passed down. And okay, let's go to number 4. Example, disease related to nervous system and skin cancer. These are all mutation in somatic cells, caused by mutation in somatic cells. Now, mutation in gametes causes inherited diseases such as thalassemia, hemophilia, and albinism, all right, which involve gene mutations, where there are changes in the base sequence of the DNA. So the chromosome will carry this abnormal gene, and this gene can be passed on down to the offspring, right? So it can be inherited because the mutation is in the gametes too. Okay, from the diploid parent cells. Then when the gametes are formed, this mutation can be passed down to the gametes in the chromosomes. Now, mutation in somatic cells. Mutation occurs only in one part of the body, for example, in the skin or in other organs that have this cancer, okay, that have cancer. So, but mutation in gametes occurs in all the body cells. For example, mutation in gametes can cause the mutation to occur in all the body cells of the offspring concerned. Okay, for example, in Down syndrome child, all the body cells, somatic cells, have 47 chromosomes with 3 chromosomes number 21. So lastly, let's test ourselves and answer this question, which is a popular question for SPM. The diagram shows an individual who has a genetic disorder caused by nondisjunction. State the genetic disorder of this girl and state the chromosomal number. So, genetic disorder caused by nondisjunction, right? And then you see the features of the girl. This is a girl who has Down syndrome, right? And the chromosomal number is 47 chromosomes, or 45 plus XX. Because it's not 44, it's 45 because of the extra chromosome 21. Okay? Now, state 3 characteristics of this genetic disorder. So we have to know at least 3 or more characteristics. Flat nose, broad forehead, slanted eyes, protruding tongue, stunted growth, and mental retardation. Okay, write in full, write a full sentence. 3 characteristics are. Okay, next, explain how this individual is born with this genetic disorder. So, okay, take note. How are we going to explain it concisely? Okay, and not too long, right? If it comes out in structured question, 3 marks. All right, this is what you can write. During Anaphase 1 of meiosis in the parent, spindle fibers fail to form. Thus, the homologous chromosomes number 21 fail to separate to opposite poles due to the phenomenon called nondisjunction. So, an abnormal gamete with 24 chromosomes, or with 2 chromosomes 21, is formed because the 2 homologous chromosomes 21 move to the same pole. Okay. Now, if this gamete undergoes fertilization with a normal gamete that has 23 chromosomes, a Down syndrome child with 47 chromosomes is born. The child will have 3 chromosomes 21. That's why we call it trisomy 21. Right, that's all for this lesson. I hope you have benefited from it, and all the best in your coming exams. Goodbye. Stay calm and study.